Okay. Thank you. Thank you, everybody. In this talk, I will present some new model, new interpretation of the Hopfield model. I will start with a very fast introduction about neural network as a complex system. Then I will explain the Hopfield model, and I will give you some new results. So what are neural networks? Basically, they are graphs, so a collection of nodes and age. Each node represents a neuron and can be in two different states, +1 or -1, if the neuron is active or is inactive. And the weight of the age can be seen as a synaptic efficiency between two neurons. So we can think our system as a couple, sigma and j, where sigma is a vector of length n, and j is the n times n matrix that contain the synaptic efficiency between the neurons. And we have to think that these two variables are different time scale. So the evolution of the j's are very slow, so we can think it as a rearrangement of the landscape, while the sigma are fast, so the evolution of the sigma are like the motion on this landscape. And we can think our output, the correct output of the network, as a configuration in this phase that we can call sigma star. So we have to do two things, essentially. The first thing is to set a good landscape, so to set the matrix J, such that the minima sigma star correspond to the task accomplishment. And then we have to choose a dynamic rule such that the system can spontaneously relax up to sigma star. So the question is that we can have more than one minima. So the question is how many minima we can have and how accessible they are. And we can think about it in a statistical mechanics perspective, thinking our cost function as a thermodynamic potential and the minima configuration as the equilibrium configuration. So this is a complex system because we can have a lot of minima, and this is good and bad at the same time because it is possible to make the system able to give several correct answers, but at the same time it is difficult to manage, and if we have too many minima, we can the network can also have a blackout scenario where the network can't work at all. So in this context, we can find the Hopfield model that take inspiration from neurobiological evidence. So the ability of human brain to remember person or object starting from incomplete information or information that are quite similar to the one that we want to recognize. And this is called associated memory. And we want to build a network that can work as an associated memory. So we want to give to a network corrupted information, and we want that the network could be able to reconstruct the correct one. So we are thinking that this network already learned, and and these memories we will call it pattern, that are essentially a vector of length n, which each entry can be +1 or -1 with equal probability. And we want to construct our network in such a way that the correct pattern will be the minima configuration, and the other point will be the corrupted pattern or completely random pattern. And to do this, a good recipe is the Ebb learning rule that tell us essentially that the matrix J is the scalar product between the pattern. Then we have to decide a dynamic rule for the neuron that is the sine of two contributions. The first one is the field acting on each neuron at time t, and then we have a stochastic part where z is a random variable, a Gaussian random variable, and t is a pseudo temperature that can modulate the noise. And it can be proved, but I will not go into detail, that this network will converge to equilibrium measure that is in this form, where H is the cost function or the Hamiltonian of the system. And we can rearrange this Hamiltonian with some algebra and write it in terms of the magnetization that will be one of the order parameter. And this is essentially the overlap between the pattern and the neural configuration. A very important parameter for the dynamics is the number of stored pattern. If we have the number of stored pattern that is proportional to n, we have a more complicated scenario, and we don't have a very rigorous result. But we can understand the dynamics looking the phase portrait of the system. We have to define before this value here that is essentially the ratio between P and N, that is the load of the system. And we have three different phases. The one that happen when T is large, we have a lot of noise, so the system is completely ergodic. Then we have this blue part here, where the system works well and works as an associated memory. And there is this huge part here, the red one, that is where complexity prevails, and we found a mixture of stored pattern. And I want to stress this point here that occurs when T is equal to 0, that is alpha equal to 0.14, that is the upper bound of the load. And why this? Because in the late 80s, Elizabeth Garden proved by general result that the upper bound for a network with symmetrical interaction as the Hopfield one is alpha equal to 1. So the 0.14 predicted by Hopfield is rather poor, and there were a lot of studies trying to improve this number here. And in this direction, we can find the dreaming neural network that take inspiration from biological studies. In particular, there are some studies that tell us that during the sleep section, we have two different phases that we continuously alternate. The REM phase that is used to remove the unimportant information, and the slow wave phase that we use to stabilize memories. And so why don't build a neural network that can do something like that? And the idea is to change the matrix J of the Hopfield model. I will not go into detail, but at the end we have this formulation of the matrix J, where T now is the time of sleep, the sleeping time, and C is a correlation matrix. And if we put this one into the Hopfield Hamiltonian, we will have a new Hamiltonian that takes this form here. And from this one, we can do, I will not go into detail here, but we can write the pressure of the system, and then by standard variational principle, we can obtain the self-consistency equation of the model in terms of the order parameter. With this, we are able now to plot the phase portrait of the new model that change with the sleeping time. So at the beginning, T equal to 0, we have the Hopfield model. And then, when the times grows, we will see that the yellow part and the blue part becomes bigger and bigger, while the red one at the end collapses completely. So at the end, we have a perfect neural network that is Eiger disorder, but this is inevitable because we have noise, and also Eiger working as an associated memory. And I want to stress that here we will find alpha equal to 1 that is predicted by Gardner, and this is also possible to prove it in a more analytical way with this theorem.
So you gave us a rule for the evolution of the J's this time.
Yeah.
Where did that come from?
Yeah, yes, the same that of the Hopfield model. yeah, it's like with that type of evolution, we can reach the minimum configuration. We are in the
So it's
basic.
not biologically inspired, it's just it's a good rule to have this.
I think yes. I'm not sure really. So at the end, we have this neural network here, and then I will try to explain you what I'm doing right now. But this is a work in progress, so I don't know if. The idea is to extend this model by putting the 0 right here and an additional bias. So this have essentially two reasons. The first one is for mimicking, because the previous model, all the neurons were used to retrieve one pattern. But now, if we think the one corresponding to null entries could be used to further activities. So it's like try to do a network with multitask behavior. And this one has a biological meaning, because at the end there are some study that tells us that our cerebral cortex, the activity is really low. So at the end we have more -1 than +1. So think of the hypothesis that the variable can take the +1 and -1 with equal probability is not so realistic. So we can add this bias here to modulate this probability. And at the end we have to change the matrix J of the system, but at the end after some trial, the good thing is to change the correlation matrix of the previous model. And I tried with different matrix, but at the end I think this is the best one. And why this? Because it can be proved that the theorem that I showed you before, the one that tells us that we can reach alpha equal to 1, is also valid in this case, but obviously with some constraint on dilution and bias. The thing is that this is the plot of the C norm, and in this part here, the theorem, we can reach alpha equal to 1, while in the blue part, the theorem is not valid. But the thing is that the part where it doesn't work is for high value of dilution, so it's okay. And the good thing is that if we increase alpha, this zone will be bigger and bigger. So at the end we have a system that works well, I think. And that's all.
Thank you.
Correctly, J's and sigmas, they evolve at the same time scale?
The J's and the sigma, no. The J's like if you change J, if this is the landscape, if you change J, you have a different landscape, like this. While the sigma is like you are moving on this landscape.
For every change of J, you let the system relax.
Yeah, but it's like you fix J, so you fix the landscape, and then you let the sigma evolve.
Was there another question? I had one in the last slide.
Yeah.
You were not.
Yeah, sure.
You were saying learning and consolidation in the title, but in which sense are we learning here? It's memorization.
It's like because it's learning like when we have memory, you highlight the good one. It's like this learning,
Like
because
yeah,
it's in this meaning, but you have already learned something. And if you go here, you can see this here, but like just you have two contribution. The j here that is for the consolidation, and minus j squared that is for remotion. The idea at the end is to delete the mixture state. So for this reason,
Okay, thanks.
thank you.
Can you try to put it out again?
Is it okay? Yes,
Oh,
this is
yes.
on.
Could you take this? Just this. Oh, so this you can put it in your pocket. And this you can attach it to your this here. So like this. I mean, I don't have a pocket. So.
So the next talk is Nagelhair, interpretable representations of RBMs by adversarial chain.
So I have a pointer.
I would recommend this one.
This one, okay. So, okay, thanks. So, so I will begin by first, thanks of all for inviting me here and this for organizing this fantastic school. And okay, so I will begin to try to formulate the question that I'm trying to, the problem that I'm trying to address. First, by just giving a quick introduction to RBMs. So a restricted Postman machine is a particular kind of a network where you have two kinds of nodes, the visible nodes, which are the ones that should model the data, and the hidden nodes which learn a latent representation of your data. And then you allow only interactions between the hidden layer and the visible layer. So what you do is you write down an energy function for the configurations of this network, which has terms that involve only the visible units, terms involving only the hidden units, and then an interaction term that couples units from the hidden to the visible layer. And then you write down the probability for the configurations of the machine, which is just the Boltzmann distribution using this energy function. And then this distribution will allow you to obtain a distribution which should match your data distribution by marginalizing over the latent variables. So essentially, this is how the RBM works. You train the RBM typically by doing a maximum likelihood. So you write, so you have some data which consists of observations of the configurations of the visible layer, and then you maximize the log likelihood of the model on these observations. So which is essentially just this term here. If you write the gradient of this, you can do a gradient ascent, but the gradient will involve, as typical with Boltzmann machine, will involve some moments of the that you can compute from the data, and then some moments that will require sampling from the RBM, which can be difficult to compute because it has an exponential number of possible states. So what you do is you estimate these moments by Monte Carlo, which in particular for the RBM you can exploit the bipartite structure of the network of the machine, and you can see that if you condition on the hidden state, the visible states are very easy to sample because they are factorized, and similarly for the hidden states given the visible states. So you can do a Gibbs sampling where you construct a chain, sampling one layer and then the other and then the other, and in this way you obtain samples that allow you to estimate this moment. And this is known as contrastive divergence, and this is how you typically sample, how you typically train this kind of machine. Now, the kind of data I will be using as an example for the uses of this machine is protein multiple sequence alignments of protein families. So a protein family is a set of proteins from different organisms which are related in an have some common evolutionary origin, and typically they share some function in different organisms, and therefore you see this reflected in that there are some conservation patterns in the sequences of these proteins. So for instance, if you have an active site in an enzyme, you expect that the residues related to this location on the protein will have some conservation in the amino acids that appear at this position. And you also expect that, for instance, if you have contacts in the structure, you will see some correlation between different positions in the sequence alignment. So now, to connect this to the RBM, I will simply look at these sites as the possible configurations, the visible units of the machine. Okay. So it has been this kind of machine has been used to model protein data, this kind of multiple sequence alignment, and it has been very useful to obtain representations where you can see that the hidden units actually learn interesting features of the proteins that you're modeling. So for instance, you can see that the patterns that the hidden units learn, so the weights that a given hidden unit learns, are usually related to, for instance, contacts between different structural contacts between different sites in the protein, or for instance, or active sites or whatever. And on the other side, the activation of a hidden unit conditioning on the given sequences, you can see that it also learns about different features of the protein. So for instance, in this case, we have a protein family where the color corresponds to different locations of the protein in the cell, and you can see that the activation of a given hidden unit or of two given hidden units clusterize the sequences according to has some relation to the sequence, but it's not perfect. Okay, has a relation to the location of the protein in the cell, but it's not.
Can you say that location, physical location?
So in this case, for instance, the protein can be located in the mitochondria, in the chloroplast, in different locations of the cell. So how can we actually see a bit more detail of how the latent representation is actually encoding a given feature of the data set? So now we consider a labeled data set where we again have the configurations of the visible layer, so protein sequences. And then the label is telling me something about the given protein. For instance, the location, as in the previous example, or can be whatever, I don't know. So what we do is we train the RBM by maximum likelihood as before. So we train an RBM. And then we train a classifier on top of the RBM that looks at the hidden layer and tries to predict the label. So if the classifier succeeds or not, will tell me whether the latent representation that the RBM learned also learned about this feature or not. Okay?
So in the RPM, the hidden layer, it's not like the variables are deterministic, it's a probability distribution.
Yeah, so what I do is I take one configuration, I sample one hidden configuration, and I send that to the classifier to see if it is able to predict from the, yeah. And then the classifier is also trained by maximum likelihood. So this would be this process. So I take one visible configuration, sample a hidden configuration from the machine, and then this will be the likelihood for the given prediction of the label. Okay. So as an example, one can ask one of the simplest questions one can ask for protein is whether a protein is from eukaryotic or bacterial origin. Okay. So here are a bunch of protein families selected simply because they have similar amounts of bacterial and eukaryotic sequences and large amounts. And what you see is that if you train an RBM on these families, and then train a classifier that looks at the hidden configuration, you see that for most of these families, you obtain a very good classification. So this is a ROC curve. So you obtain a very good classification between eukaryotic and bacterial sequences. So it means that the machine in supervised training is automatically learning about this particular feature of the protein. However, this has the following problem that so if you look at the weights of the classifier, so how the classifier is looking at the hidden units, so in this case you take each hidden unit and you see the sum of the weights that are coming into it. What you see is that the classifier has to look essentially at all the hidden units, so it's not localized. So it's not like you have one hidden unit which is coding for this. So the term that people use in machine learning to refer to this kind of situation is that the representation has an entangled is learning about this feature in an entangled way. So all the hidden units are somehow coding for the feature, but there is no single hidden unit that is learning about this feature. Okay. And this means?
Sorry, I'm a little lost here. Imagine that I don't care about RBM. You want to do this task, distinguishing catalytic and bacterial. You could have built directly a classifier
Yeah.
on the visible unit.
Yes.
And would it be better or worse? Or?
It would be almost as good, actually, if you compare. But the question is, what we want to do is simply, the task that we want to do is not actually to distinguish between this kind, is to improve, is to understand whether the representation learned by the RBM is learning about this feature and to disentangle it or not, to improve this. So what happens is that the RBM is learning about the feature, but it is entangled across all hidden units in the machine. Okay. So the question is, can we improve the interpretability of these latent representations, which means that we do something so that, for instance, only one of the hidden units is capturing the information about the given feature. So what we can do, so we use the same architecture as before. So we have the RBM, we have a classifier that is looking at the hidden units, but now we train them both at the same time. So in this case, what we want to do as a first example is to actually erase this information from the hidden representation completely. So how we would do that? So we train the RBM and the classifier simultaneously, but in an adversarial way. So which means that we are maximizing the log likelihood of the data in the RBM, and at the same time we are penalizing the performance of the classifier on the hidden representation to be able to predict about this feature. Okay, with a given weight that penalizes this performance. So if we do that, we train this way. So simply what we are doing, if you maximize, you take this maximum inside here, simply what we are penalizing is the mutual information between the latent representation and the label that I'm looking at. Okay. So you can see that we are using this classifier simply as a way to estimate this mutual information. It's one way of looking at it. So we do this adversarial training, which simply means that we do this gradient ascent, descent. So where we update first the weights of the RBM to maximize, to minimize, and the other term to maximize this other term. So if we do that, first, in order to, so there is an issue here which is related to taking the gradient of the adversarial of the classifier with respect to the weights of the RBM. So because there is in the middle, we have this sampling of the hidden units. Here we have to use this same reparametrization trick mentioned before. So to see this, simply if we look at the classifier objective, it will depend on parameters related to the weights of the classifier, and it will depend on the weights of the RBM because it is looking at the hidden layer of the RBM. Okay. So this is essentially the objective function that we have to optimize for the classifier. However, in order to backpropagate the gradients from the RBM to this classifier, what we can do is that we can write the hidden states as a function of an extra random variable, the weights of the RBM, and the visible layer configuration. So the introduction of this extra random variable, which does not depend on the weights of the RBM or the configuration of the visible layer, allows us to take the gradient of this hidden configuration. So for example, if we have Gaussian hidden units, we could write them like this. So where this is the mean determined by the RBM, this is the standard deviation also determined by the RBM in some way, and this is simply a standard random variable which does not depend on the parameters of the model, and then it is easy to take the gradient of this with respect to this weight. So, and this allows to take this gradient in a way that is by sampling. Okay. So if we do that, then we train simultaneously. So we see that we improve the log likelihood of so in this case the pseudo likelihood, which we use as an approximation of the RBM looking at the data, and at the same time we are penalizing this is will be the loss function for the classifier, so the cross entropy, okay, for the classifier. So we are doing these two objectives at the same time. So this one should go down and this one should go up. So at the end of this training process, we can inspect whether the classifier looking at the hidden units is able to predict the label that we are looking at or not. And we see that in fact, before this area under the curve, so this measure of the performance was 0.9, now is brought down to 0.6, for instance, in this case. and this, of course, will depend on the value of the alpha, so of the weight of the penalty that we impose. As we increase, this will decrease more the ability of the classifier to look at the. So we are in fact erasing this information from the latter representation, and this comes also it has some impact on the log likelihood of that the RBM is able to. So on the quality of the log likelihood of the RBM of the given data. So as we erase this information, the log likelihood gets worse. Okay, so it means that the machine is really losing an information that is relevant for the data that it is modeling. So the next thing we can do now is to have a classifier that looks at not at all the hidden units, but leaves one hidden unit out. So what this means is that we will erase the information from most hidden units except one of them. Okay. So if we do that, we do the same idea, then the selected, all these hidden units will lose the information about the label, and this hidden unit will actually be able to, will learn a useful representation of the label of interest, which in this case is this division between bacterial and eukaryotic sequences. Okay, so this histogram is simply the activation of all the hidden units. So these are random hidden units, and this is the one that I selected. And what we see is that there is a useful division between for eukaryotic sequences and between bacterial sequences. So actually, in this case, we managed to concentrate this information on one of the hidden units. Okay. And actually, so if we look at a classifier that is trained a posteriori on the latent representation, if the classifier looks at all the hidden units, or if it looks at the selected hidden units that if it looks at only the hidden unit that I selected, it will be able to predict the label. Whereas if it looks at all the other hidden units, it will not be able to predict the label. Okay. Yes, this is the last slide. So the idea is that with this we have a generative model where we can actually, by tuning the activation of this particular hidden unit, we can generate sequences which, in this case, for instance, correspond to bacterial or eukaryotic sequences, but we can do the same with other kinds of labels, like the specificity of an enzyme or whatever. So that's it. Okay, so I finish here. And this is this one. But, thanks.
When they're changing, there is one question, but no hard questions. So.
So the question is, so when you put the classifier, it seems that you are kind of doing some.
Sorry?
When you put the classifier, it seems that you are kind of putting interaction between the units. So what happens if you try to put lateral inhibition between different units? And I mean, is there a way to bring the classifier to lateral inhibition?
Yeah, but what that will do is that you will obtain sparse representations, no? So you can try to sparsify the representation, but that does not guarantee that for the label that you care about, which I mean can be one label, can be multiple labels, that you will concentrate this information of one of the hidden units, because there is no reason why it should be like that, right?
Okay. Thank you.
So in the pockets.
And last speaker of today is Moshir. Yeah, I remember I didn't copy the whole time for internal program. Plain cells emergence and learning in invariant data with RBM, breaking and dynamical restoration of continuous symmetries in the weight space.
Thank you for saying it out. Okay, so welcome to the talk. I'm hungry as well. Please bear with me for 15 minutes. Okay, I'll start quickly with an RBM, even though my predecessor just explained it. So you have visible units where you put data, and then you couple it with some latent variables called hidden units. And this was also, it's a generative model, so it's something Max Welling was also explaining in the morning. And these are connected by some weights, so Wi mu. And the feature about this is there is no connection between any hidden unit or any visible unit, but each hidden unit fully sees this, and so it's fully connected across the layer. And it's defined by the distribution, so 1 over z e to the power minus vh, and the energy function is defined pairwise, so VI, HI coupled with some weight W mu I and some fields, some BI biasing fields on these variables, and then you have some potentials on these hidden units. So here you would put typically activations like ReLU or Gaussian potentials, things like that, in the machine learning sense. And if you marginalize over this P V of H, if you marginalize over H, you get a distribution that looks like 1 over z exponential of some BVI plus the log Laplace transform associated with this input. So I mu is the input that each visible unit sees. So basically the point is, even though the joint distribution is simple, it's pairwise between V and H, but the marginal over H, so PV in this machine is not pairwise, and it's usually, so if you take binary values for these for VIs, so each VI and HI can take binary values, then this is typically of the form of log cos hyperbolic or something. So you can learn higher order interactions between the visible units. And that's the power of RBM. And we train them using mini batch stochastic gradient descent with something called persistent contrastive divergence with K steps. Okay. So the first thing, I'll quickly move to results. So the first thing we did was train this RBM on a simple Ising model, 1D Ising model on a ring, so periodic boundary conditions. And this RBM only had one hidden unit. So each visible unit can take plus minus 1. You only have one hidden unit. So I would like to, if anyone has a guess of what the weights would look like after training. With just one hidden unit, so the weight is just one vector of as many spins you have.
I don't understand what your task is.
Sorry?
I don't understand what your task is. What are you trying to train for?
I am trying to make the RBM learn the Ising model distribution. So the form that the Hamiltonian is some VI plus 1. So we are trying to see if the RBM matches this distribution that comes from Ising model. So any guesses on how the weight would look like? It's just a simple machine of weight is just vector. So one option would be, since, sorry, please. Yeah, so since there is a symmetry here, you can move the spins about and nothing changes in the distribution. So one thing would be all the weights are uniform. So all the weights are uniform. That's one thing. Second thing could be the RBM just does something like the PCA and it learns the top eigenvector. But none of it happens. The weights after training look something like this. So they show, so this is the visible unit index. I have used 100 Ising spins, and this machine was shown 10,000 configurations of these Ising spins from this. And this is the distribution of the weights after you finish the training. And you see this has a peak structure at some position. Now the next thing I do is I change the the temperature of the correlation length of this model, and I plot the width of this peak as a function. And you see that the width of the peak is increasing. So this very simple hidden RBM has actually learned to coarse grain this data distribution at the relevant scale. This very, very simple model. So you are seeing an increasing correlation length. If you have multiple hidden units, so here I'm showing an example for 3 hidden units, you see peaks which are well separated along this manifold. And they have slightly less width, and as you increase the number of peaks, so here I can even go up to 100 hidden units, then the peaks are really, really small.
So you told
Yes.
the system that it was nearest neighbor?
Sorry?
You had told the system that it was nearest neighbor?
No, the system doesn't know anything about it. The simple is just, it just does this energy function. That's it. It knows nothing else about the distribution of the data. Now, but there is a problem. This has some sort of symmetry. You can move this around and nothing changes. So in the loss, nothing changes. But here you see that this peak is fixed at some point. But what point is this? If the model has this translation symmetry, which point should I fix it at? So what we also realize is this machine dynamically restores this symmetry. If you keep training the machine, if you go longer in the time scale of training, here I'm plotting, this is also results from RBM with one hidden unit. I'm plotting the center of the peak as a function of number of training epochs, and this is just a repetition for 10 different experiments. So you see the center of these peaks, or these peaks actually diffuse on this ring. And if you just go ahead and measure the diffusion constant, it's almost 0.5 spin square per epoch. Something I should say here is that to see this diffusion properly, you need a lot of training examples. The landscape should be smooth enough such that with small SGD, so small learning rate, you can see this diffusion. If you increase the learning rate and you have less data, you would destroy the peak structure. So it's a compromise between, you have to increase the number of data you're showing. Now this is from an RBM with 3 hidden units again, and you see the correlated motion. So there's the center of the peaks again as a function of training time. You see that the 3 peaks that I showed before diffuse in a correlated fashion. So there is interaction between these hidden units, and they try to occupy as much as the manifold as they can. So mutually exclusive places, and they try to maximize the information. So this is what I showed. In weight space, you have a scenario like this. You have these weights which are limited or restricted to this manifold, and they diffuse along this manifold. In this high dimensional space, there exists a low dimensional manifold because of the symmetry of the data along which the weights diffuse. Okay, going ahead. Yes. Does this always happen? So does this peak always emerge? And the answer is no. If you have beta, so beta is the inverse temperature from the model. If the beta is very small, you realize that if you plot the absolute value of sum of weights, so you realize that the machine till this point does not learn anything. The weights are essentially very noisy and they sum up to an average 0. But if you cross a threshold, then you start learning things. Then you see this emergence of the peak. And the same if you plot n over s, which is the system size over the number of training examples over the system size. You see if the number of training examples are very few, you again don't learn anything. And then when you only increase the number of data, that you start learning something. And this is not because the SGD cannot find it. It's if you look at the correlation matrix of the sample, so and you plot the top eigenvalue, you see there is a transition almost like the MP spectrum. So you only see this after the top eigenvalue leaves the bulk. Essentially, before that, if you have very few data or very weak signal, then it's dominated by noise, sampling noise. Okay, now we move to another model. So the XY model, so you have cos theta I minus theta I plus 1. What I will do is I will discretize this model as a Potts model. So now VI, the visible units can take values from 0 to p minus 1. And the energy function is now a kernel which mimics the cosine. So it's really the label minus the next label. So it's just the discrete version of this. Again, so now the visible units can take p values. I will use p as 10 in this, and the same as hidden units. And now the weight weight vectors is not just a vector, but each weight has 10 components associated to it, because there are 10 Potts states here. This model has an invariance, so has for any integers k and l, you can sort of move them around on the lattice and change the theta as well, and it doesn't change anything. Now when you train the RBM, this is the results you see. So this is when you have this is with ReLU, and this is with normal Gaussian potential. And so what you see is the same visible unit index I'm plotting, but now each weight has 10 states. So what essentially seeing is, again, there is some localization at some point in space, but now there's also localization in angle. So if I go ahead, so what I'm plotting here now, if I go at this point where the absolute sum of the weights is the highest, that's what I am plotting, and I look at these 10 points. So, and this is a fit to a cosine function. Mind you, the machine does not know anything about a cosine function being there. The machine has learned this cosine function. This is a fit. And again, if I take the sum of this along this direction, and I plot here, so you see a localization in space. And so if I fit cosine again, this is the phase of the cosine, which is constant in some arbitrary value. The phase is in some arbitrary direction in this sort of space. And the frequency, which is also constant along this part of the space. And the frequency is 1 over 10, because we have 10 Potts states. So the machine has recovered the correct frequency. So in a way, loosely, we can say that when the machine has learned from this distribution, the machine essentially looks at one position in space and in one arbitrary direction. So Going ahead, if you have multiple hidden units in the same, you see that they are mutually separated along the along space position, and in angular position they also have different phases. So they try to maximize as much information as they can. There is very strong cross inhibition between these units. And now you see a similar phase transition that I showed before, but now there is something more interesting because you have two thresholds. So if you look at these low n over s values, you haven't learned anything. You have essentially 0, but now when you start going to higher values, you don't see a peak structure here, and essentially the weights get some value that is non-zero, but they still don't point anywhere. But here you see that in the angular space, they do learn a phase and the cosine. So phase 1 threshold has been crossed, but the other not. And now then you also cross another threshold and you see this localization in space. Okay, so I'm going to talk about why is this relevant. So high dimensional inputs or data have underlying low dimensional invariances. This is something called the manifold hypothesis, which many people I support. And it's important to understand how the statistical structure of input stimuli, so what we see, what data we hear, or what sort of images we get, shapes its encoding by the brain areas and further higher processing. The place fields, so these fields by the hidden units are what I call place fields, are general features observed in retinas, in place cells in the hippocampus. I'll quickly talk about place cells in the next slide. And also as optimal solution in Gardner's theory for single layer neural networks for translationally invariant data, which is essentially what we had. And you can find this in this paper in which they show even in online optimization, the place fields for translationally invariant data cover the whole manifold, and this is the optimal solution, having place fields. And this representation is actually very useful because it can be this place field representation, it can be learned by recurrent neural networks, and it can allow storage of multiple maps. With the same few cells, you can store multiple and retrieve multiple maps using the same cells. This is what happens in our brain as well. Quickly, something about place cells. So we have cells in our, for people who don't know, we have place cells in our brain which tell us essentially where we are. Like if I'm standing here right now, there's a cell in my brain that's firing, that telling me I'm here. If I go outside, it will not fire. It will, and another cell will fire, telling me that I'm there in that position. So for this rat, here, whatever cell is fired has different colors. That's shown here. I'll take 1 more minute. So what this Joffrey lab, what they do is they make a mouse run on a helix. So it is essentially a helix going up. And so this is the recording of the place cells when the mouse is going up, and here when it's going down. And what they show is they've sort of unraveled each sort of turn of the helix on a map. And essentially what they show is, because of this, so in this experiment they haven't changed the pitch, but here what you see is the localization is along angles. So when the mouse is at specific angles, these cells fire, but there is no localization along space. The cells fire in almost all, wherever in height the mouse is. And the mouse therefore would not definitely know which height is on. If the helix probably had a higher pitch, then we might see localization in space as well. So you see sort of this sort of transition, the phase transition that I described before. Anyways, so conclusion first, condensing everything into 12 minutes is challenging. We try to understand how symmetries affect the representation of data without explicitly giving the architecture the symmetry, what CNNs do. Learning is accompanied by symmetry breaking, and it's dynamically restored. This is something reminiscent of continuous attractors, but in weight space. Usually CAs are taken in the activity space. And the phase transition, so after a critical value, and it's tempting to sort of connect these things, but we have to be careful because we don't know a lot of what happens in the brain, but it's pretty incredible that unsupervised learning rules with maximum likelihood learning gives the major features about what happens in brain as well, cross inhibition and formation of receptive field. Thank you.
Thank you. One, two, three.
You mentioned diffusion, these peaks.
Yes.
Is it pretty diffusion? Check that? Or
I mean, in a sense, if you
does
plot,
does it move like round motion? Or?
so the way I checked it was I plotted the mean square displacement as a function of time, and
It's kind of linear.
it's kind of linear. The thing is, the landscape has small irregularities, and you really need to go to a large amount of data to remove it.
So, thank you to all the speakers. Please catch them at the dinner, after dinner, tomorrow. They're all here, right? And I'm sure they will